It is my absolute honor to introduce you to our audience today. Dr. Dhanisha Jhaveri holds a joint appointment at Master Research Institute and the Queensland Brain Institute, University of Queensland, and is a Master Foundation Research Senior Fellow. The focus of our laboratory is to understand neuroplasticity mechanisms, particularly those underlying the production and integration of new neurons in the adult brain, and their roles in regulating circuits implicated in mood and cognitive disorders. Dr. Dhanisha has several awards to her name, including the prestigious Indian National Science Academy's Medal for Young Scientist of the Year and an Australian patent. She's passionate about mentoring young investigators and believes that quality mentorship is vital for scientific success. Today, the title of her talk is Stimulating Neural Stem Cells for Brain Repair and Regeneration. Regenerative medicine and the amazing reparative potential of brain lies at the core and the heart of my objectives and my goals, and I'm pretty sure that everybody, aspiring biologists, medical students, neuroscientists across the world, when they were in high schools or in the university, they have thought about this, like neural stem cells. Like, that's very amazing, and I'm very excited to listen to you today. Thank you to be here, Dr. Dhanisha.
Thanks. Should I share my screen?
Yes, definitely.
Yeah.
Audiences, please note, I'm adding a note in the chat box. You can add your questions using Q, and you can also comment using C.
Just 1 second. I want to see. Can you see the presentation?
Yes, we can see.
Is it in the presentation mode or?
No, it's not in the presentation mode currently.
Okay. Now?
Light show.
Is that better?
See, yeah, it's better. It's right.
Yeah.
Yes, it's fine.
Okay, excellent. Thank you, Pranjal, and thank you, Stuti and Team Stimulus and Project Encaphalon for your kind invitation. I think I really need to congratulate you for this fantastic initiative, which I'm really privileged to be part of. I mean, I really find the vision and the mission of your these two projects very, very encouraging and also commendable in terms of trying to instill and really foster the scientific inquiry in neuroscience. And it's really amazing and wonderful to see that many of these projects and things are really led by the students. So I wish you all the best for both Team Stimulus and Project Encaphalon. And as you would have heard from Professor Shubatole, I mean, really, neuroscience is, I think, at a very, very exciting juncture. It is really a frontier that we need to understand. It's an endeavor to really understand basic fundamental principles of how we learn, form memories, feel emotions. And I think being on that path of that journey of discovering some of these fundamental mechanisms and the brain connectivity and how that actually outputs into behavior are all aspects of neuroscience, which I'm sure many of you will go on in future to embark on. So as Pranjal said, that in my lab, we are really interested in understanding the mechanisms which drive the production and integration of new neurons in the adult brain, and really understand what role these neurons play in regulating some of the cognitive and emotional functions. And this all with a really bigger vision or bigger view of harnessing this form of plasticity to basically help repair or regenerate some aspects of the brain or parts of the brain that may be sort of compromised in neurodegenerative or neuropsychiatric conditions. But before I jump into really the core of my research and our findings, I would like to actually highlight my journey, because I think I also started out very much with a very basic curiosity of what does our brain really do? And I actually started out doing my bachelor's in science, in life sciences and biochemistry at St. Xavier's, Mumbai. And I was really captivated by some of the teachers there in terms of how they instilled in me some of the scientific inquiries that I still carry on to this date. Following my bachelor's, I did my master's in zoology, and that was really driven at that point in terms of trying to understand how an embryo develops. Really, I was fascinated, and the first book that I had read was this Triumph of the Embryo from Louis Wolpert. If any one of you have read it, would agree with me. It's a fantastic book in terms of how it lays down that really the organism that forms from an egg and a sperm is the whole process of the molecular cellular orchestration is really a miracle. And for those of you who haven't read it, I certainly urge you to read it. So after my master's, I basically got into, as Shubha just mentioned, at the Tata Institute of Fundamental Research, and where I pursued my PhD in the field of developmental neurobiology. And this was really working on fruit flies, on Drosophila, working out during pupal development, how really the olfactory circuit develops, especially the circuit which is related to the sense of smell, in terms of how the antenna actually sense the sensory neurons in the antenna, how they actually connect into the brain, and what are the exon guidance molecules that regulate this process. And it's really during this time that I got fascinated by the prospect of understanding really the production of new neurons in the adult brain. And a postdoctoral fellowship that I received really got me onto the plane to Australia and in Brisbane, where I spent a number of years doing my postdoc work. And now, since about 4.5 years, I lead my own independent research group here. So that's really about my journey. But in this journey, I really want to, again, take this opportunity to acknowledge the enormous sort of mentorship that I have received from a number of individuals who have really instilled in me the scientific rigor in terms of asking the right questions, having the broad vision, and also importantly, really the compassion towards understanding people, but also understanding and fostering how to realize one's full potential. So I don't want to go and name every one of them, but certainly my PhD mentor, Professor Veronica Rodriguez, was absolutely instrumental in basically making a scientist out of me. So I'm incredibly grateful to her. So now coming to Brisbane. So this is an aerial shot of the University of Queensland. And you can see that there is a bridge across this river. And I have the privilege of actually working across two institutes, one which is on one side of the river and the other on the other side of the river, which is the Queensland Brain Institute and the Mater Research Institute. Queensland Brain Institute is a purpose-built neuroscience institute, which houses about 450 to 500 people. We have about 44 different groups who basically focus on different aspects, from molecular, cellular, circuit behavior, and in terms of sensory to other neuroscience aspects. And at Translational Research Institute, we actually endeavor to do some of this translational work in aspects of neuroscience, but also in other areas like cancer and immunology. So coming back to my the talk, in terms of really, it was the Spanish neuroscientist and the father of modern neuroscience, Santiago Ramón y Cajal, in early 1900s, when he was basically drawing all the the images or what he saw under the microscope in terms of the neurons, and that supported the neuron doctrine. What he thought when he was drawing all these connectivity was, he basically said that in the adult centers, the nerve paths are something fixed, ended, and immutable, and everything may die and nothing may be regenerated. Given that this requires an exquisite connectivity between neurons, he found it really hard to imagine how there could be additional neurons that could be added into the circuit without really disrupting the circuit. And that really led to the dogma of that perhaps there are no new neurons generated in the adult brain. Whatever we are born with or during early postnatal development, what we generate is what we will have, and there can be only death, but nothing new. But now we appreciate really the extent of plasticity that occurs in the brain. In fact, this plasticity is, and this ability to mutate, so this mutability is in fact essential for carrying many of the functions in terms of encoding novel information or association, changing behaviors, etc. And there are many forms of plasticity mechanisms that have been now uncovered, going from epigenetic modifications, which lead to gene expression changes, from there to synaptic strength in terms of which are activity driven, so activity dependent changes in synaptic strength, or in terms of even circuit tuning. However, a very interesting and a striking form of structural plasticity that we now have come to appreciate and has been very firmly established is this de novo generation of new neurons. Not only generation, but their integration into the existing circuitry. And much of what I'm going to talk to you about today is all about how do we really generate these new neurons, the regulation of this process, and what are the functional implications of generating new neurons in the adult brain. So this process, which we now know, is called the adult neurogenesis. But this process, to give a historical perspective, was not something that was always believed. In fact, for much of the in the 1900s, for the first almost 50 years, the dogma of proposed by Ramon y Cajal existed. And it was only in about 1960s that a very talented scientist at MIT really challenged that dogma. And this is an important aspect because I think as a scientist, it's always important to remember that while dogmas exist or while the concepts exist, it is equally important to think about it and sometimes if we have the evidence to challenge those dogmas. So Joseph Altman really challenged the dogma of no new neurons and provided the first hint of this process occurring in the adult rat. And this is some of the sort of the timeline that I have put on in terms of which highlights some of the important sort of discoveries. So even though he proposed and he showed quite nicely that new neurons are generated in the adult hippocampus of the rat in 1960s, that discovery was really met with lots of skepticism. In fact, majority of the neuroscientists at that time completely discounted his discovery, so much so that after a few years, he was forced in a way to actually retire from science. He actually quit science. So this is what I highlighted here in red. So this period of about 30 years, people really didn't believe it. And this was mainly because there was really a big figure at that time, Pascal Rakish, who basically refuted this claim and said that in the adult primate brain, really he didn't see any evidence of new neurons, new neurons production. And that was perhaps because of the limitation of some of the approaches and maybe the model that he used as well. But there was a resurgence of interest in this field in early 1990s, and that was really led by two groups. One in Australia, Professor Perry Bartlett, with whom I did my postdoctoral fellowship, and in Canada, which was led by Ronald Brent Reynolds and Samuel Weiss's group. And they showed for the first time an isolated, in fact, precursor cells in the adult brain. They showed that there are precursor cells that are present in the adult brain that could have the potential to generate new neurons. And these were studies done in culture, in vitro, not really in vivo. And since then, in early 2000, there was really an explosion in this field in terms of gaining a detailed understanding of what this process is and what is it really required for. And I was particularly interested, and I was particularly excited by this finding of René Hen's group, which showed that some of the behavioral effects of antidepressants actually required adult hippocampal neurogenesis. And I will come to that later in my talk. So today I would like to actually just highlight some of the discoveries that we have made and give you snippets of kind of work that we are currently pursuing in our lab in Australia. So you have heard quite nicely from Shubhatole about the hippocampus. So in the brain, hippocampus is certainly a major area where adult neurogenesis occurs. And it is an area which is critically involved in our learning, in forming memories, and also regulating mood. And there is also another area, which is the subventricular zone, where, again, the production of neurons continues in the adult brain. And these neurons that are generated in the subventricular zone, in fact, migrate into the olfactory bulb. So these are the two major neurogenic areas that are known in the adult brain. But as I will come to later, there are perhaps other areas which are less studied, where production of new neurons may still continue in the adult brain. So if you look at a hippocampus or a brain of a mouse, and this is where in the lab we conduct most of our studies using. So we use mouse as a model system. And if you take a cross section of that hippocampus, you can see the dentate gyrus, which is really the V-shaped structure, and the CA3 and CA1 areas. And it's within this dentate gyrus, there is a layer which is known as the subgranular zone, where these neural stem cells or precursor cells really reside. And again, in the lab, we have a number of ways to visualize these. And one that I have shown you here, which is the neural stem cells, which are labeled here in red. So you can see that quite nicely that, and this is using a mouse line which expresses a reporter gene, which is driven by a proneural gene, which is expressed only in the neural stem cells. But if you leave these cells long enough, and in this case, we left these mice for 4 weeks, we can see that these stem cells actually divide and form these new neurons, which you can see quite beautifully in terms of how they extend the dendrites into the molecular layer and also send axons into the CA3 region. So this process of neurogenesis, which is basically the production, the differentiation into neurons, their survival, and their ultimate integration, is this multi-stage process of adult neurogenesis. And many studies have implicated this process in regulating aspects of learning and memory, and also in regulating mood. Importantly, this process is really sensitive to a number of environmental stimulations, such as physical exercise, as I mentioned, antidepressants, environmental enrichment, all of these actually have a positive impact whereby they increase the production of new neurons or increase neurogenesis. And as well, there are negative regulators of this process, such as stress, aging, inflammation, which leads to either impairment or dysfunctional neurogenesis. So in the lab, we are really driven by asking some important key questions in this field, and which are really related to what are the mechanisms regulating this neural stem precursor cell activity and neurogenesis. So really at this stage, how really neural stem cells integrate information from extrinsic and intrinsic signals, how adult-born neurons contribute to local circuitry and impact behavior, because these neurons they receive inputs from different areas of the brain, mainly from the entorhinal cortex, but also from cholinergic innervation from the medial septum, and in terms of how really these neurons integrate all these signals and then lead to an output that regulates behavior. And ultimately, by understanding the first two steps, can we really harness this process to improve mood or cognition in any of the neuropsychiatric or neurodegenerative conditions? So these are really the big questions that we are asking in the lab. And some of my initial work was really focused around understanding really how antidepressants regulate neurogenesis. So at this time, when I started my postdoctoral research, it was already known that antidepressants actually enhances neurogenesis, but the mechanism by which this was happening was really not clear. So one of the first things I did was really set up this new assay, which I called the slice sphere assay, where I grew or I cultured hippocampal slices which are the organotypic slices, and basically treated these organotypic slices with different antidepressants from different classes, both the selective serotonin reuptake inhibitor class and also from the selective norepinephrine reuptake inhibitor class. And what I did was I treated these slices with antidepressants, and then after a period of about 7 days, dissociated these slices, made them into single cells, and these cells, I subjected them to mitogens. With the premise that the cells which have the stem cell or the precursor cell capacity would actually proliferate and give me these balls of cells, which we call neurosphere. So in a way, each neurosphere is coming from basically a single precursor cells. And I would count such neurospheres in the dish to see what effect my treatment had. And interestingly, what I saw was things like Prozac, fluoxetine here, another selective serotonin reuptake inhibitor, citalopram, really didn't have any effect on really changing the precursor cell activity. As opposed to that, what I found was the norepinephrine reuptake inhibitors really robustly and significantly activated these precursor cells. And this was really exciting because this is for the first time that one could show that not all antidepressants acted in a similar fashion in terms of regulating neurogenesis. There were the class of antidepressants which could actually stimulate the precursor cells activity to increase the production of new neurons. And this basically led me to think more about what was the underlying mechanism and the principles. And what we really figured out, and this was doing, again, these neurospheres assay that I just described, in terms of only the precursor cells can actually form these balls of cells called neurospheres. And in presence of neuronal depolarization, or particularly in my case, presence of norepinephrine, directly could lead to activation of these cells. And what this meant was that I had many more these spheres, but also that when I added these two together, I almost got an additive or a synergistic effect. So this experiment told me two things. One, that first of all, I was activating something that was otherwise quiescent or dormant. So this was the first demonstration in a way of a neurotransmitter activating a dormant population of stem cells, which would not normally respond to just mitogens. It required additional signals. And in this case, we figured out that it was norepinephrine. And after that, we have actually a number of other factors which we have discovered also leads to the activation of these cells. But importantly, it also showed that different stimuli could perhaps activate different population. So this experiment really showed me that maybe we are not talking about a single population, but perhaps there are distinct neural stem cell populations which reside in the hippocampus, which can be activated by different stimuli. And this has obviously enormous implication. And this was the hypothesis that I tested in my next study, where I set out to really identify and isolate and purify these stem cell population. So this is one of the transgenic mouse line that we use. This is reporter GFP, green fluorescent protein, which is driven by nestin. Nestin is considered one of the stem cell markers. And here we have really the complexity of these neural stem cell in terms of how they have really very intricate arborizations that are spanning the granule cell layer of the hippocampus. So we combined this mouse line with another marker in terms of a fluorescently tagged epidermal growth factor. And we basically isolated this double positive population, which constitute a very small percentage of the total hippocampal cells. And you can see what these cells look like. So these are the nestin GFP positive and epidermal growth factor receptor positive cells. And when we cultured these cells, so this was done using flow cytometry, a method where we can put in cells in the sorter, and we can ask the sorter to basically sort out cells which are green in this case and red as well. And it'll give you cells which are green and red both, or just green alone without red, or no green or no red. And what we can do after that is we can take these cells and plate them at clonal density. So there is only one cell in a well. And by doing that, we could show quite nicely that norepinephrine and potassium chloride, which mimics basically depolarization, could activate this stem cell directly. So what we found was majority, almost 85% of the stem cell activity was present within this population. And this population responded both to norepinephrine and potassium, such that, again, addition of both these factors led to an additive effect. So basically, this led us to the model of maybe there are, perhaps at the moment, as we know, two different subpopulation of stem cells. There could be more, which could be activated by different stimuli. One, which we have shown later on, could be activated by LTP. And in this case, we showed that norepinephrine population, responsive population, really required the activity of the beta-3 adrenergic receptors, which we showed was both sufficient and necessary to stimulate this population. And we also showed that these two different distinct populations really are not homogeneously distributed along the hippocampal axis. In fact, the potassium responsive population is present much more in the temporal part of the hippocampus, whereas the norepinephrine, you find majority of them in the septal part of the hippocampus. And at the moment, we have the theory that perhaps these two populations may play, they may generate neurons which may play slightly different role, maybe one more related to spatial learning and memory, and one related to mood. And this is very much an ongoing project in the lab. And we have found additional factors that can regulate these two different populations. But what's really exciting from our purification studies, where we were able to purify and isolate these studies, it really gave us this unprecedented opportunity to start really inquiring what these stem cells really are in terms of what is their molecular signature, what kind of genes they express, and can we actually start to unpick and unravel novel regulators of these stem cell activity. So that's exactly what we did. So we did a transcriptome analysis on this, and we found a number of genes, about 1,000 genes, which were which defined the signature of these neural stem cells compared to the rest of the cells that are there in the hippocampus. And expectedly, we found a number of cell cycle genes because, these are proliferating cells, a lot of plasma membrane genes. But what was very interesting for us was this observation that we found that there are many extracellular matrix related genes which were expressed in the stem cells. So extracellular matrix, for those of you who don't know, actually is made up of sort of glycosaminoglycans and some of the proteins, which constitute about 20% of the brain volume. However, the function of extracellular matrix is not very clear, although there are papers implicating the role of extracellular matrix both during development of the brain, but also in terms of more plasticity mechanisms, which are involving maybe synaptic pruning and likes of that. So as I said, this extracellular matrix is really a matrix which is outside the cell. It is composed of different proteins which are held together by some basically carbohydrate moieties in terms of sugars. And what we found was that indeed some of the extracellular matrix associated factors such as hyaluronic acid is actually, in fact, indeed present around these stem cells. And this is a study that actually was a PhD project of one of my students, Saurabh Bandhavkar, who was in fact recently finishing up and writing his thesis. And what he found was that if we digested this hyaluronic acid, it basically enhanced the production of new neurons. And I'm not going to go into the details of all the various steps that we have found are by which this increased production takes place, but it suffices to say that if you put in the enzyme which digests this hyaluronic acid directly into the hippocampus. Saurabh did these what are called stereotactic surgeries in the adult brain, where the mouse is kept under a stereotactic frame, and you open up the skull and drill a little hole, and actually through a syringe inject this hyaluronidase, the enzyme that breaks up this hyaluronic acid in the hippocampus. And then we can actually label the proliferating cells by giving mouse a thymidine analog, which is bromodeoxyuridine. And we can do histology, immunohistochemistry, and show that these red cells are the ones which are the proliferating cells, and green cells here are really the immature or the new neurons. And you can count how many such cells do we get, and both in terms of hyaluronidase increases the total number of these immature newborn neurons, double coatin cells, and you can see that indeed there are many more these proliferating newborn neurons in this treatment group. I'll not go into the details, but he went on to show that this was because he was able to activate the stem cell population directly by digesting this hyaluronic acid. And he found the receptor on the stem cell which mediates this activation. So there are a number of receptors for hyaluronic acid, and he found one of them which is expressed by the stem cells, which when we block it, prevents this activation. But I just want to give you an example of the kind of work that we do in terms of not only we have identified this mechanism, but we wanted to ask the question whether this mechanism can actually have a functional consequence. So what Saurabh chose to do was use a task where we can actually assess spatial learning in mouse. So this is a task which is called the active place avoidance task. And what happens is that mouse is kept on this rotating arena. and in a room. And this room has multiple cues. And they're quite distinct cues. And mouse actually uses these spatially distinct cues to figure out where is this invisible shock zone. So depicted here in red is an invisible shock zone, where if mice enters into that zone, it will receive a very mild shock. But over a period of days, mouse will learn that this is not the zone to enter. And the mouse does it by just utilizing these different spatial cues. So we asked the question that what is the consequence of increasing these newborn neurons? Does it really have an effect on the ability of the mouse to learn in the spatial environment? And what Saurabh found was indeed it does. So as I was just telling you, that mouse actually learns to associate this shock zone with respect to the spatial cues. And you can see that with number of trials on different days, the mouse is receiving less and less number of shocks. So it is really learning. This is a good learning curve that we get, and depicted in red are the controls. But depicted in the black are the controls, and shown in the red are the ones where Saurabh had injected or digested this hyaluronic acid in the hippocampus. And what he found was those mice where he had injected hyaluronic acid, they showed an improved or an accelerated learning. You can see the difference really at day 2 and day 3. So if you compare the number of shocks that individual mouse receives on day 2 versus day 3, you can see the hyaluronidase treated one, they basically, all of them received significantly less number of shocks on day 3 compared to the control mice, which was still not learning. So even though control, they learn at the end of 6 days, these mice actually had an accelerated learning. So this was very exciting, and he has gone on to show that this accelerated learning was indeed dependent on these newborn neurons, because we have a genetic tool now where we can selectively kill off these newborn neurons, and we could show that this beneficial effect of hyaluronidase in terms of improving learning actually goes away. So that's for the one of the discoveries that have come from basically understanding and after purifying the stem cells, where we have been able to unravel a new mechanism, in this case one which is regulated by a simple carbohydrate which interacts directly with the stem cell through a cognate receptor, which can then lead to the activation of otherwise a quiescent population of precursor cell, increasing the production of new neurons, which has a functional consequence. But I told you that we are also interested in understanding how really these neurons contribute to the local circuitry and impact behavior. And in that particularly, we are interested in understanding really what role these newborn neurons play in regulating aspects of mood. Now, I must mention that, in this process of this generating new neurons, there are different stages. And this is really this immature stage, which is what we are most interested in, because really it is at this stage that these neurons are quite distinct and different from rest of the neurons in the hippocampus. Because at this stage, these neurons actually they have very high intrinsic excitability, such that they can be excited with very low amount of current. They actually show very high degree of synaptic plasticity. And it's also during this period where there is an extensive dendritic morphology and dendritic development that is going on. So it's really this phase which I have sort of highlighted here, which we believe plays an important role in regulating some of the aspects of both mood and cognitive regulation. And based on these different properties, in fact, almost about 2 decades ago, a neurogenic theory of mood disorders was proposed, which basically stated that stress leads to impaired neurogenesis or reduces neurogenesis, and antidepressants, which basically sorry, the stress which impairs neurogenesis is responsible or leads to a low mood or dysregulation there. And antidepressant, when because they enhance neurogenesis or restore neurogenesis, it leads to basically restoration of the mood. However, this theory, while has been tested by multiple labs, we really still do not have a clear understanding, a mechanistic understanding of how really newborn neurons contribute to mood regulation. And this was something that we had basically embarked on as well. As we all know, that there are obviously genetic factors and environmental factors such as stressful events, which are both play a sort of a interactive and a synergistic role in regulating some of this neuroplasticity mechanisms, which make in this case, a mouse or a human susceptible or resilient to mood disorders such as anxiety or depression. And hippocampus is really an important region of the brain which regulates the stress response. And this is via the hypothalamic pituitary adrenal gland axis, which I'm sure many of you are very much well aware of. So what we actually did, and this was really the work of my another PhD student who has just recently graduated, Mei Zhao, was to combine the stress model and combine it with our ability to deplete or ablate these immature neurons. So we have this transgenic mouse line, which expresses a diphtherotoxin receptor under the promoter of this gene called double cotin, which is expressed by the immature neurons. So when you give mouse diphtherotoxin, it selectively kills off just these immature newborn neurons. And you can see that quite nicely here. So these are the DT treated, where you see many less of these green, which are the double cotin expressing cells. And we can quantify that here as well. And again, what May did here was to give these mice either just normal water or water which had the stress hormone, corticosterone, over a period of 4 weeks, and ask the question, what happens to anxiety or depression related behavior in these mice if the newborn neurons are now ablated? And you can see quite nicely here that we can efficiently and significantly reduce these newborn neurons in both the vehicle treated and the co-treated group. And the test we use is called the novelty suppressed feeding test. Now, this is a very interesting approach avoidance kind of test, which is classically now been used in mouse and in rodent studies, where the animals are food deprived for a period of maybe about 18 to 24 hours, and then they are put into a novel chamber. And when they are in the novel brightly lit chamber, in the center of the chamber is where we put the food. And the amount of time, so the mice basically have this motivation to go and eat food because they're hungry. But at the same time, they are in this novel, brightly lit, anxiogenic environment. So amount of time that the mouse actually takes, the latency to start feeding, is considered a measure of anxiety. And what we can see quite nicely is that the vehicle treated mice, they begin eating quite rapidly. They exhibit low levels of anxiety. The ones which have been treated with the stress hormone show high levels of anxiety as expected. But interestingly, if you ablate the mice where we depleted these immature neurons in the hippocampus, by itself didn't have an effect, but in combination with the stress hormone, in fact, completely prevented the anxiogenic effects of corticosterone. And this was really an exciting and a new finding, where we think that actually stress leads to a dysfunctional neurogenesis. So not only these neurons that are born during stress period are probably not integrating or forming properly. And we have done a number of studies to actually show that it actually stress changes their physiology, so electrophysiological properties and their connectivity into the circuit such that they are actually short circuiting. So if you remove them from that circuit, in fact, you restore the behavior. So these findings are really, really exciting, and this is the work that we are currently finishing and writing up. So in the end, I just want to spend and tell you about one more findings that we have had, and that is related to the fact that I initially mentioned. Even though hippocampus and subventricular zone are considered the two major neurogenic areas, there is some speculation that maybe there are other areas in the brain, such as neocortex, amygdala, hypothalamus, which may also permit production of newborn neurons. And we actually had focused on one such area, amygdala, for the interest in terms of its role in regulating emotional learning and memory. And what we found was, as opposed to the hippocampus, amygdala contains precursor cells, but these are all in the active form. There are, in fact, we could not find any evidence of the quiescent population of precursor cells in the amygdala. And we could show that these precursor cells indeed do divide and form neurons. And we did that by using a number of different approaches, including some viral approaches, and showed that they actually could generate functional neurons that could fire the action potential. And these neurons had a signature of that of the interneurons. So this was the work that we published in 2018. So all in all, as I said, that neurogenesis to me is not only an important, but it's really an exciting form of neuroplasticity mechanism. And in the lab, we are really endeavoring to understand the molecular and cellular regulation of this process, how neural stem cells actually how we can regulate the activity of neural stem cells and change the production of newborn neurons using cell culture transcriptomic approaches. We have begun to study really the circuitry, how these neurons then actually integrate into the circuitry. And we have been using selective lesion, pharmacogenetic and optogenetic approaches. And eventually we want to understand how these neurons and the circuit actually alter the behavior. So with that, I would like to end and acknowledge both my present lab members, May and Saurabh, whose work I alluded to, and some of my past lab members, our collaborators who have been very generous in sharing some of the reagents, and are mainly the funding bodies, which is the National Health and Medical Research Council of Australia, Mater Foundation, and Stem Cell Australia. And I'm happy to take questions, but also I would encourage questions and happy to answer them via email.
That would be very nice. If we can have your email from your website, is that okay? Or
yeah,
if you want to share, as you feel comfortable.
Yep, I mean, I can put it on, or people can just search for me. I'm sure they will find me.
That can work. That can definitely work. Sure, that can work. I'll say from Kahal and Altman and Das's paper to neurogenic theory of mood, cognitive and mood disorders, that was very nice talk. And we have a question over there actually. So in the neurogenic theory, does neurogenesis affect mood by effects on learning and memory, or could it be something else altogether? So it is asked by Annapurna.
Okay, this is a very interesting and important question. And there are multiple theories on this, depending on the task that one uses in terms of behavior. In our hands, using the model of stress that we have in terms of this stress hormone via the delivery through their drinking water, we in fact find that it doesn't really affect their spatial learning capacity. So their spatial learning is not affected, but their anxiety-related behavior is indeed affected. But having said that, if one uses another or a different task, there is always a possibility that one may find differences in cognitive abilities. So I would say this: the jury is still in terms of not out there, and this is still a very much an open question.
Okay, I hope that clears Annapurna. So there is another question. I guess he's a bit confused. Hyaluronic acid stimulates stem cell regeneration directly, and how come using sorry, I cannot. It is a bit of a
So
tongue twister to
hyaluronidase,
pronounce.
yeah.
Hyaluronidase stimulates stem cell regeneration at same time. So he's a bit confused. Hyaluronic acid stimulates stem cell regeneration, or does the enzyme stimulate
No,
the stem cell
so
regeneration?
yes, sorry for the confusion. It's not actually the it's basically the the digestion of hyaluronic acid. And I forgot to mention that actually hyaluronic acid comes in different flavors. So you can have really high molecular weight hyaluronic acid, which are really long chains of these disaccharides that you can cleave off to form a low molecular weight hyaluronic acid. And what we have found is, in fact, depending on the size of the hyaluronic acid, you can actually have opposite effects. So the low molecular weight stimulates or activates the stem cells, whereas the high molecular weight keeps them in the quiescent stage. And I didn't have time today to go into all that data, but in fact, hyaluronic acid is playing this dual and distinct role. And what hyaluronidase, the enzyme that digests it does is actually it breaks down, and we have evidence for that, breaks down the high molecular weight into low molecular weight, and that leads to the activation of the stem cells.
So in regard to the same study, I have a question. So the mice takes time to like learn the things, right? Because according to me, the adult neurogenesis, it takes at least 6 months. So it's kind of a heartbreak. I understand, like antidepressants could not work for 6 months, and it's kind of heartbreak for me. How much time does the mice take to learn when there is a hyaluronidase acid and it's stimulating stem cell regeneration?
Yes, so I would like to actually correct you in terms of in the mouse. Actually, it's not 6 months. In fact,
Okay.
it is actually between 2 to 6 weeks. So these
Oh, that's
neurons actually start functioning at 2 weeks itself, and by the time they mature to basically grow to 6 weeks, they are almost indistinguishable from their mature granule cell counterpart. So and it's a good question in terms of how quickly these mice start to learn, and it's usually between that 3 and 4 weeks when these neurons are really in their immature stage, where increasing their numbers, actually the mice performed much better.
So there's a stark difference between rodents and humans because the time difference is so high, 6 months and 3 to 4 weeks. We should do something about humans as well to study. So we have
Absolutely.
an
But
yeah,
as you can imagine, studying this process in human is not that straightforward. It's not as
right,
straightforward as studying hematopoietic stem cells, where most of us would volunteer to give 10 ml of our blood, right? Who
right,
is
right.
volunteering to give me a part of the brain?
Exactly. Yeah, that's
But
right.
having said that, in fact, we have actually just recently set up a clinical study in
Okay,
which
that's nice.
what happens is the patients who have epilepsy, who are refractory to normal medication, have to undergo the resection of the temporal lobe. And we are actually approved now to get a part of their hippocampus that comes out as a part of their surgery procedure. And we are going to look for stem cells, especially these dormant or quiescent stem cells in the human brain using those samples.
That sounds really interesting. So I have a next question here. Could you please elaborate or comment a little more regarding the incorporation of new neurons into the neural circuitry?
Okay, so I don't know exactly what, in terms of, there are many aspects of new neurons being incorporated into the neural circuitry, both in terms of the receiving their appropriate inputs that are coming from different cortical regions and driving their output into the CA3 region. So new neurons mainly connect, and in fact almost exclusively connect to the CA3 region, but also along the way they can connect to some of the interneurons. So this process of how this integration happens, what are the different molecular and other factors that regulate this process is something that we still do not completely understand. But it suffices to say that as much as increasing the production is interesting and important, equally important, or perhaps even more important, is understanding their integration. And what we have found is that during the stress treatment, in fact, these neurons don't integrate properly, and therefore they behave in a dysfunctional fashion, such that when you remove them, you actually basically can restore or ameliorate the changes in anxiety. So I think this area, in terms of how newborn neurons integrate into the circuitry and how they drive the feed forward and the feedback inhibition, is going to be very, very exciting areas of future investigation.
Definitely. I would like to go to the last question. We have many more questions, and I encourage you to please message Dhanisha or mail Dhanisha. So the last question is about the statistical analysis done during anxiety and depression study in mice after restraining them from food for a while. So there was the study, the second last study. Ritz wants to know about the statistical analysis you performed over there. I'm not really
Okay, so
clear on
we
what.
actually, because this was of 2 by 2 groups, in terms of we had the vehicle and the core treatment with and without ablating newborn neurons. So we actually used two-way ANOVA to do the statistical analysis, and then compared basically post hoc using Bonferroni tests. So I can go into the detail if they want to ask me, and in terms of I don't have on top of my head what the variance is, but that, as you could see there, it was pretty tight.
Thank you so much. I think so. We are at the end of the talk. And please, guys, I'm so sorry I'm not able to take your questions because we don't have time left. Please mail Dhanisha. Thank you so much, Dhanisha. And it was a very exciting talk. Actually, I'm a big fan of Kahal, and I just clicked to the talk. So I'm really excited. Thank you so much, Dhanisha.
Thank you so much, and I wish you all the best. I mean, this seems to be a very exciting lineup of speakers and program for this weekend and the next weekend. If I'll try and join in as much as I can, but other than that, wishing you all the best and good luck with everything.
Thank you so much.
Thank you.